Okay, so today's class is I guess it's one of the most important throughout the lecture. So we want to focus really. So let's discuss about optical microscopy. And I will actually go through a little bit of the applications. So that's pathology and light microscopy. So that why we are doing this course is eventually we want to apply for the pathology and many interesting fundamental biology applications. So light scattering with the tissue for disease diagnostics is possible, and it's a very, very interesting application. And I want to re-emphasize that, biomedical optics by what itself is saying, hey, biomedical is an emerging field because we are facing a world with aging society and more of a degenerative diseases are prevalent. And, when we are living longer, we will have suffering from more of a disease. And optics is a very new and field which provides a very interesting solution for this. Okay, so that's why I think it's very exciting, and still there are many, many interesting areas are awaiting for us. So let me introduce the microscopy, which has a long history, and I want to discuss histopathology, which means histo means a study of tissue. I mean, biological tissue, and pathology. And histopathology is basically looking under the microscope, seeing that a sectioned tissue by a pathologist. So that's when your daddy or your grandparents go to the hospital saying that, oh, I got this disease, diagnosed. And so that's an imperfect gold standard. Why I put imperfect? It's a limited. And their biomedical optics may be able to help. So that's my point. And we'll discuss a very simple technique today. Before we get towards the end of this lecture, we will cover a more modern and very advanced techniques, which is actually kind of groundbreaking, because that's why there are so many Nobel Prizes coming, because of the changing of our concept. Previously, think it's impossible. That has been possible by our knowledge and application of the physics of the light and even chemistry of the light. Okay, so we'll cover color illumination, an objective lens with NA, so-called numerical aperture concept. And those who love photography, you will notice that F number in your camera lens, an immersion or condenser lens and light source. So let me start with the how do different tissue constituents scatter light? And actually it goes back to our previous discussion, and light scattering and light interaction with the tissue, and how can we utilize it for disease diagnostics. So the point is, we discussed the light interaction with the tissue. There comes a scattering event that depends on the relative size of the light wavelength versus the object, which is a scatterer. And between them, there will be a different kind of scattering properties. And what if those interacting entity is now biological, cells and subcellular organelles? And outside of the cell, there's a bunch of extracellular proteins that will scatter light. And the organelles will be different in cells when the cell status is in healthy, normal versus unhealthy disease status. So that catch may produce meaningful differences of light scattering property. So that changes in scattering and even absorptions will be due to changes in the organelles and the cell product. For example, normal cell versus cancer cell, we know a bunch of the differences in their morphology, and they may affect the light scattering properties. So that changes in scattering may be also due to changes in local microenvironment. So, even with the bare eye, we can sometimes see bruise, which I got this bruise last night by falling down from a bike, and I got a bruise. And what's happening? Some cells are dying, necrosis, and it's reddish. That means there's an inflammation process going. And inflammation is very good for us because it tries to heal, and wound healing is happening. And in some cases, angiogenesis. There is a good aspect of angiogenesis and bad aspect of angiogenesis, such as in cancer. But when you run, you practice and exercise hard, your heart will make a more microcapillaries. That's a good aspect of angiogenesis. We can even talk about therapeutic angiogenesis. But these will eventually change in scattering of the light, and you may want to utilize it. So thus, optical scattering may be important for disease diagnostics. And how the scattering is different in different microenvironment, fundamentally in optical point of view, there will be the refractive, local refractive index changes. So that change matters. So the bigger delta refractive index, the more scattering happens. And of course, the scatter sizes, we know that from previous knowledge, tiny particles, so-called Rayleigh scattering, is more like isotropic, but the bigger particles, which is comparable or bigger than wavelength, we call it Mie scattering, and that scatters more. So the bigger, the more scattering. And also the geometry matters, the amount and direction of scattering. You remember that we can utilize the cell distinguishing by the forward versus side scattering. The side scattering is more when the scatterers are more of a complicated shape, such as more lobular shape, more nuclei, different shapes will create more side scattering. With that information, we can even distinguish between different white blood cell type, monocytes having one single cell type, the other kind of a white blood cell, granulocytes, and other white blood cell having multiple lobules will increase the side scattering. So that gives a very precious information. And also in some kind of tissue, interestingly, we can even utilize the tissue's basic birefringent property. And what that means is this tissue is kind of anisotropically organized, that the direction of polarization actually will have a different scattering, so that this asymmetric shapes makes the changes in birefringence, and you can utilize it for optical detection and diagnosis. Let me give you an example. So for example, like your colon tissue having a multiple layer, and with that, it has a very specific organization. When there's a colon cancer or tumor arises, what happens is these collagen layers and structure of these surface layers are disrupted. And this is a very good target to use a polarization sensitive imaging, actually show quite different contrast. So that's something I want to kind of remind you why this biomedical optics can be very interesting, a good tool. So let's look into the pathology. And pathology is literally what it means, is study of a disease process. And in the laboratory pathologist, looking at the tissue specimen, and that has been the gold standard for disease diagnosis. But more problem is you need to take out the tissue, okay? So pathology usually are having two categories. One is anatomical pathology and the other is clinical pathology. So anatomical is histopathology. As I say, histo means a tissue, pathology is a disease. Looking at the tissue structure, you find out what kind of diseases. So for example, surgical pathology in the surgical operation room, you cut off those suspicious part of the tissue out, and you look at under the microscope the cells, and you focus on the architecture so that you can be able to find the margin of the tumor so that you can carve out the tumor only, not too much for the normal tissue, and also staging of the disease, because the proper staging leads you different type of treatment and also giving different prognosis for the disease progression for the patient. On the other side, you can also have a cytology. Cyto means cell, so you don't care about the nearby structure. You just take out the cells spread out. So cells without architecture. For example, for a female cervical cancer diagnosis, the doctors will have a tool like such as Q-tip to scrape off the cervix and then spreads out onto this microscope slides. That means you only want to look at the cell, okay? So that's called pap smear to detect whether someone has a likelihood of cervical cancer or not. Or even like sputum, like you're like saliva, not only saliva, the tare, like that sputum also smear you can watch under the microscope. Of course, you do this, you won't be able to see anything because cells are usually transparent. So you do need some kind of giving contrast, such as some kind of dye staining. And a famous one you have to know is immunohistochemistry, or shortly IHC. What it means is histo, and there's chemistry and immuno. So what it means is you use the specificity of antigen-antibody interaction to figure out very specifically the molecule of your interest. So detecting antigens in cells or tissue section. That's based on the shape of the cell or tissue architecture that we call anatomical, while the other hand, you can actually just collect the blood from the patient and you can do the pathology. That's called clinical and laboratory tests of analyzing the body fluids such as blood, urine, and tissue. So one example is on the right side, CBC, or completed blood count. You can have this blood and you can have blood count based on this like flow cytometry type plot. You can actually measure whether you have too many white blood cell indicative of whether you are in infection or not. Okay. So all of this, in fact, the light is almost always used. Okay, so let's then go back to the microscopy from the history of our like human. Let's go back to like now it's 2000, so 400 years ago, in Netherlands, there's a Van Leeuwenhoek who actually made the first recorded simple magnifier microscope. So that looks like this. It's a little confusing to look at it first, but if you look at closely, there's a one lens. So that's the microscope. But it's a very tiny, so you need a place the sample. So you need to have a holder, and you need to move the sample this, let's say, X direction, and it's already aligned, I guess. And then you need to move ups and down to make a focus. Okay, so sample translator. And this works well enough that he started looking at many things. In fact, the vocation of Van Leeuwenhoek, he is a seller for the like this fabric. So he need to see the thread better. So that's how we actually made. At the time, there are a bunch of lenses are available at the time. And one of his kind of student is Johann Ham. Actually, he actually observed human sperm first. So like you're curious, you know that there's a sperm, what's happening inside the sperm? So there's a sperm, you know that. There's a sperm which will create a human being later on. So he made this assumed that this picture have a small, tiny human being inside the sperm. That's that case. But that's because they have a poor resolution. They only depend on their imagination. So that's more like human being inside. That's called, let me see, Sadaf. Can you guess what this is called?
Homunculus.
Very good. Yeah, it's called homunculus. You know, humans are called Homo sapiens, So a human being, a small tiny ones are inside. Homunculus is also used when you are learning neuroscience in our cortical areas of brain, which has all the parts of our body parts is a sensing, and that we also have homunculus. Right, now afterwards, so with single lens, you develop further, like that's like Robert Hooke, you also heard about Robert Hooke, who actually defined the word cell for the first time, okay? And he made this simple compound microscope. And what that means is compound means you are now multiple lenses, not a single lens. So it will be a better performance, but you need to shine tiny area with a high intense. So you can see lamp and water flask as a lens and to shine this. So sample illuminating lamp device is still good. And then afterwards, 1930, Carl Zeiss, as a popular microscope. That's why Germany is still like really strong in terms of microscopy. And you can see this one's almost now like close to 90, 80, 90 years old one is still pretty much similar to and usable today. So it's a functional, you can see eyepiece. As I said, there's objective lens, there's a body tube, okay? You know, remember you have a 4F system tube lens, and there's a stage, and there's a condenser lens, which like illuminates the sample in a small area, and there's a mirror. Now we replace this part with an LED, right? Because it's much cheaper and easier. So I want to bring your attention for like, what does your pathologist, the doctor do at most of the time? They use this microscope as a tool, and most of the time, over 90%, they use this H&E stained tissue. So you can see I put this typical magnification of 2x, 10x, and 40x. And remember that about 40x is typically you can watch the sample without having immersion oil or water. So it's much more easier. So you can see, if it's a 40x, you can see the cell nucleus more clearly. And these are, I guess it's mucin producing goblet cell. So 10x, you can see cells somehow. 20x is very good, typical high resolution microscope, and 40x. And Jungmyo, can you guess what kind of lens is here?
Condenser lens.
Very good. You are condensing the illumination light onto your sample. Okay, so as we go to discuss histopathology one more time, and that means examination of tissue to study the manifestation of disease. And typically you remember that light tissue interaction, the mean free path of the light is about 100 micron. What that means is you don't want to have that more thick sample to have a perfectly nice image because of prevention of multiple scattering event. That's why you have a thin sample, very thin, only a couple of micron, and you stain H&E, and you get tissue, look at tissue under the microscope, look at architecture, morphology, and pattern recognition. So this one is actually my sample. When I was a postdoc, I was doing colon cancer research. So these are the colon cancer model from mouse. And look at these, the colon pits are a little bit irregular, and you see the nucleus are like bigger and the morphology is not clean. So these area of arrows indicate there is something going on, a tumor process. And the other thing I want to emphasize is histology stains. Without contrast, we cannot see and analyze anything. So a typical one like this pinkish one is called H&E, more precisely hematoxylin and eosin. And I realized I made this on opposite in previous lectures, okay? Because hematoxylin stains nucleus, which is an acid. You know, DNA is deoxynucleic acid, okay? Acid stain, and that makes it blue. It doesn't look very blue depending on the color, but it's like bluish, and so it's darker for the nucleus, while E is a base stain, which usually stains a protein, extracellular matrix, whatever which is non-DNA part is pinkish, right? So with these contrasts, our pathologists are expert looking at this and tell all the story of what's going on. While all this is converting into AI will taking over, because to train one pathologist, it'll take years of training and cost, and AI can do the job with not sleeping and with very fast way. So we are actually in an era of fastly changing times for this expertise. And H&E is great, but sometimes you want to see more specific molecules. That's where comes special stains. So whatever it is not H&E, you want to see specific, that's special stain. For example, I want to see collagen. Okay, collagen distribution changes. Then there's most of the time a special stain for that. For example, it's a Mason trichrome stain, staining collagen. Also, you are interested in specific molecule or protein in tissue, then you use antibody, and so the immunohistochemistry. And I have to say, this is more like an art, that in BMSC, we have a number of laboratories which can do this. This is not easy, I have to say, and very expensive, but whatever you see this type of image, it looks like a dark brown things, and these are immunohistochemistry, because these are antibody staining, usually with the horseshoe, I forgot about that. Horseshoe reddish peroxidase. So it's a brown pigment. I'll show you more example. So, however, this is standard of the care, and why optics is trying to conquer this area, because when we do this, let's say I'm suspicious about I have a esophageal cancer. So I go to a doctor, and they will go through my esophagus. They pick up like this. One side they take off like 90 degrees, and another layer they take off the sample, They send the tissue to make this stain, and then pathologists look at it. Hey, I cannot find anything, and so you're fine. But 5 years later, I will find I have a big esophageal cancer. Then I can tell the doctor, hey, you guys did the wrong job. Why? Because so far the gold standard is only sampling, and very tiny amount part of my esophagus, there's a huge mistake can happen. Okay, so if it's not invasive, we could actually virtually go through every single layer part of my esophagus, so I'll be much easier to find out if there's any problem. And another thing is making this sample always involved with an artifact, because you have to embed the tissue into, let's say, paraffin or OCT, and when you cut the sample and you are putting in chemicals, so there's a shrinkage issue, and the most importantly later on, you need to go to find a good doctor, which will not likely to miss your problem. So inter-observer variability. These final ones may be resolved by adopting AI machine learning technique. Okay, I just want to show you one example of immunohistochemistry so that you can recognize. So immunohistochemistry shows this type of images, negative, equivocal, amplified. You see this dark brown. This is where what we are interested. For example, this is a HER2 staining. HER2 is a growth factor, sometimes overexpressed in breast cancer tissue. That's indicating a poor prognosis. So you want to figure out that's kind of a expression level by this way. And also sometimes you are seeing this fish or fluorescent in situ hybridization, which you can see that these blue ones are nucleus, but look at this dark red one is actually HER2 expression, but some of this one have a double one, which is indicative of amplification in the gene chromosome. There's a multiple spots of HER2 gene. So that's why this one gives a amplified expression. So this is kind of a gold standard, and you want to figure out the disease types based on that. So now I want to go back to our bright field microscope, which is the most simplest and wildlife, and typical microscopes are just looking at the absorbance because you are looking at the color of the light. So this is microscope, and Jungmyo told about condenser, but I want to add one more thing here. So typically there's a halogen lamp on the bottom. There is a diaphragm over here, and that we call field lens and field diaphragm. So remember, aperture stop and field stop. So that part of illumination is conjugate onto the sample. That's why it's called the field diaphragm. Okay. So play with the real microscope at the end when you have a chance in time. Okay, now let's move on to a little modern optical microscope, which is more sophisticated. You can see a lot of electronics involved. However, the basic concept is pretty much similar to what you have already. So this transmitted light is making transmission illumination of the image, but modern microscope also has typically capability of a fluorescence. Epifluorescence micro. What that means is you have a fluorescent lamp and you have a dichroic mirror, which we'll talk later, and then it'll shine the sample from top. That's we call epi illumination, and then the fluorescence light comes back. Here, I want you to draw your attention. So in your laboratory, you'll find this looking very expensive type microscope, but you learned about 4F system. You now know what 4F system consists of: objective lens as one single lens. But there gotta be another lens before making focus onto your eye or on the sample or your CCD camera. There gotta be somewhere. Look for that. And here I want to show where is the tube lens located. You see it's collimated light, so it's 4F system. Here comes the in between your binocular and the body. You take that off, you'll find out one lens sitting there. So that distance of objective lens to this is called a F1 plus F2. And if it's back focal plane, that's called tube length, which are described later. So let's understand how microscope images are formed. So we've learned already, and you've done already the practice with a single lens. You know that 1 over A plus 1 over B equals 1 over focal length of the objective, the lens. Okay, and the typical microscope or old ones are usually having only one single lens, and so the B, the distance from lens to the image, is called tube length, and that's. US has a 160 and European is 210 millimeter. So when you find a good like kind of old microscope, you'll find this one. But modern microscope having 4F system, we call infinity corrected. Where comes this infinity? That is because objective lens, the sample object here, the light coming to the object, which is happen to be focal length, will be collimated. So that's why it's called infinity corrected. And you thus need a second lens called tube lens to make a focus. And that we call 4F system. This is great for all kinds of scanning microscopes such as confocal, two-photon microscope should have this geometry, or many fluorescence microscopes should have. Why? Fluorescence microscope need a filters. If you put a filter here, you will generate spherical aberration. That's bad for your imaging. That's why you want to put filters here, not there. In fact, one of my students' recent paper, he actually put filter over here, and so it was not the perfect position, okay? So how come we can make an image? Because you put an eyepiece there and look, actually your eye's lens now become another kind of imaging system. So you are looking at this real image onto your retina, okay? So for infinity corrected magnification, it will be like the ratio of focal length of tube length versus objective length. And also you notice that this is a minus. And one thing I want you to know is a working distance concept. That's the end of the objective tip to the object. And that's defined as working distance. Why is it important? Because you don't want to ruin your microscope objective if you don't know about this, because objective is so expensive. And here comes a little more complicated at first. So I deliver you conjugate plane concept in bright field microscope. So pay attention here. So I will describe you a two-way. One is you have illumination onto your sample, and that also goes back to your detector or eye. That's illumination. Think about illumination. Do you want to focus down your illumination onto the sample at one point? No, you want to shine your illumination uniformly onto your sample so that you can image. But think about imaging part. Imaging is basically a light scattered or out at each point of your sample. That is, as a point source, it diverges out, and that should focus back to your screen. So these two aspects you have to consider. That's why I have two on the left and right. So let me show you, explain the left part first, which is illuminating light path. So let's say your illumination comes from a lamp. So lamp, in fact, has a filament. So here is a condenser, diaphragm, condenser. Look at the condenser, there's one lens, and you shine on specimen as a parallel light. So that's very important, parallel light. And here comes a focus down from your filament. So you will, when you put your screen here, you will find out a nice filament, but that's what you do not want this one onto your sample. So that's why you focus down your illumination on the other side such that it will be parallel onto the sample. You have uniform illumination, goes to your objective lens. Let's consider objective lens as a combined one single lens that will focus down at the focal, we call it back focal plane, and that will come back to your eyepiece, and your eye, it will be spread out because your illumination has to be spread out. And there comes this 4F system of things, lens and condenser. Your filament will form over here, which is a back focal plane. And there also has another one. And that's aperture or illuminating conjugate planes. And when I say this is about illumination, however, look at how image is forming light. As I say, image is coming from the sample as a point source, it comes out, okay? And they will have objective lens, and they will form a focus, and then later they'll focus onto your eye. Okay, this is in eyepieces. So image forming, and these planes are called conjugate planes. And for that imaging plane, this is a specimen plane. And let me go back to have this diagram. You remember aperture of condenser is only kind of making the amount of light, while this part is a conjugate of your image. So that is conjugate of your sample plane. So you name this as, let's say, Kohinoor. Can you guess what kind of diaphragm is this? Think about the two types of aperture. One is a aperture stop, the other is field stop. Is it going to be aperture or field?
No, I didn't get it.
That's fine, because what I say is, you see the focus down is the same as image. So that's a field when you talk about field of view. So this limits the field of view, called field diaphragm. Okay, so this way of alignment of especially the illuminating path uniform is devised by German scholar called Queller. So this is why I call color illumination. It's uniformly illuminating the sample. So when you take off this eyepiece, you will be able to see the filaments. That's how color illumination is done. Okay, so this is an iteration of color illumination. This can be a little confusing, so I want to reiterate. So you have light filaments over here. So this is actually a little confusing. So from filaments, it focuses down filament, so it's aperture diaphragm, and it passes through condenser. Your sample is over here, uniform light onto the sample, and it back focal plane, your filament is focused down. So objective back focal plane, you will actually see filament image over here, and then it goes back to your eye, and then it spreads out because you don't want to see your filament. However, the image coming from specimen plane, it goes to objective lens and tube lens, it forms an image. And with the eyepiece lens, you will, with your eye lens, you will focus down to find out your image. So the sample plane over here, and this is called image plane, that image is here and image is there. So imaging of lamp filament is projected onto illuminating aperture diaphragm here, and parallel light is actually through the specimen is important, called color illumination for even illumination. And the specimen focal plane has to be evenly illuminated, and parallel light on the back focal plane, and here like objective lens and parallel light here of the objective lens is what's happening. So that's we call color illumination. So when practically, when you are dealing with a microscope, and this concept would be important. Now I want to go back to the objective lens again, and the concept of part focal distance. So once you have an objective lens, I just having a little highlight for something basic you better to know. So you already learned plane means a field, flat field correction. Apo means apochromatic, so chromatic aberration is corrected. The most important thing is this zoom, so lateral magnification, slash and numerical aperture. We'll deal with this, and or immersion. And this one's a little specialized called DIC microscope, which we'll talk later. Also very important, the final part here. You see infinity mark. That means this is basically infinity corrected microscope. It has to be used as a 4F system. It has to have tube length. Okay, that's why it says tube length is infinity corrected slash 0.17. This number is also important because typically your sample should have a cover slip on top. And that cover slip thickness is 0.17 millimeter or 170 micrometer. This objective is mostly corrected for that. In terms of there's a cover slip number, you should use 1.5 for this. And then as I said, working distance in millimeters 0.21, it's only 210 micron. So it's a very, very good objective lens. If the sample is almost touching, you gotta be very careful not to crash down your sample, also ruin your objective. The other concept is, I said, working distance. And when you have a nose piece, you change the objective, you will find out the image is still okay, focus. That we call parafocal. So this table is something when you are in a laboratory with multiple types of microscope, or when you are building your microscope, you should know about that, about Leica, Nikon, or Olympus, Zeiss. When you are building your imaging system, with this infinity corrected, you should use proper tube length. Nikon Leica has a little longer compared to Olympus and Zeiss. Why is it important? Because you should use this one to define your magnification correct. So instead of 200, let's say you use tube length of 100 millimeter, this one should be half, 30x imaging you are obtaining. So that's what you learn from 4F system. The magnification is the focal length of tube length divided by focal length of the objective length, and parafocal distance And also the threads are different. You should buy proper adapter from Thor Lab or anywhere, because these German ones are metric 25, Olympus is RMS, so there's always a converter. So the objective magnification is folding when you have a tube length with a matching tube length. Of course, you can use different one, but you have to realize your zoom will be different. Okay, now I want to convey the most important concept called numerical aperture, like collecting power. So we remember that this aperture stop is a physical element that limits the amount of light information. And so this theta is important. And Ernst Abbe, who defined this first time, he's a father of microscopy, by the way, and he defined the range of angles over which the system can accept the light, and that he defined sine theta. However, these mediums sometimes are not air anymore, so you put more generalized refractive index. So n sine theta is the one you have to remember, and sine theta is always less than 1 or the same, but with this refractive index, you can have numerical aperture higher than 1, which means you will have a better resolution in the end. What's the limit meaning of this? Because NA is limiting optical information. What about the other scattered light? You're losing, but this one you are collecting. So that's optical information entering into your optical imaging system, and this will will tell you the resolving power of a lens. So it determines a resolution. Let me show you here. So if you have high NA, we have point source, point becomes like this, but high NA, point becomes more sharp. So having higher resolution. So that's resolving power. That's why NA is such an important concept, actually the most important concept. So many times we want to have a larger NA, which means a larger angle. So collect more light, you can reduce your exposure time, and your image will be brighter, and you have a better resolution. On the caveat, think about your camera or professional. You have now taken pictures of focus at here, and everything blurred, like Jungmyo and Suhyun right now. It's more like digital technique, but the really out of focus is blurring, which means the depth of field is more shallower with higher NA. I will get back to that later. Okay, so I want to just show what happens with your real objective, the best one. You take a point source, you actually do not never get point. You will get this so-called airy disk. If I plot it on the like profile of intensity, it'll look like this. So there's a central ring and ringing happens. So that's why I plot this way, low, high, NA. And this is called a point slide source will be imaged as spread. So people define as a PSF or point spread function. Of course, the narrower, the higher resolution. And this is what characterizes your performance of a microscope, because it's defined as optical resolution. So how do we define optical resolution? Spatial resolution. And let's say, when we say resolve some things, okay, we know that there are two particles nearby. What if they are closer together? Oh, I can still see they are two particles. What if they are even closer? Can you say there's two particles or not? Now you get confused. This is what we call resolving power, or defining how to define resolution. So if I plot this intensity, then hey, it's hard. So maybe in between something like this, let's say minimally distance to resolve things. So mathematically, it's just Rayleigh, Sir Rayleigh, just how about, let's say first 0, when it's 0 meets the next one, then you will have this formula. this profile, and that's kind of barely resolved. So it's just easy. So I define Rayleigh criterion by Rayleigh. 2 points are distinguishable when the central maximum is at the minimum of the other. Well, that makes sense. Of course, you can define another way too, okay? But the important underlying physics for this is diffraction. That's coming from wave nature of light, why the bending of the light with this aperture, and because aperture for us is finite, and the lens circular aperture is creating this ringing of diffraction pattern called Airy pattern. And so for us in practice, you will just have a Lowe's background and the top in half. So full width at half maximum is, in practice, you report in your paper that this is your resolution. Why? Because it's very hard to find out the lowest minimum because of the background noise. Okay, I think it's now